Wow. It gets easier every time I do this stuff. Uh, okay, anyway, so here we are with a little live stream. I have been lazy and haven't done many of these lately. Eric M is here. Eric is one of my followers on Patreon. And one of the deals with Patreon is if you're a subscriber and it doesn't cost much, you can subscribe for $1 a month. That's perfectly fine. Uh, Before I do these live streams, I post the Zoom link so they can get in and contribute if they want, or if they don't want to, that's fine too. It's a little bit risky because I worry that someday some creationist is going to figure this out and they're going to sign up for my Patreon for 1 month just so they can jump in and be obnoxious in one of these videos. But not a problem here with Eric. Okay, so what do I want to talk about? Um, the thing is, the last video I did was on this topic of um, this really simple but kind of time-consuming, hard work sort of experiment that was done back in the 1980s and 1990s, where they were working with grasshoppers and fruit flies and trying to figure out what are the genes involved in pathfinding. So, this is a key feature of the development of this nervous system. You've got all these neurons scattered all over the place, and they all have to send out these axonal processes, these neurites that terminate in growth cones, and the growth cones just kind of work their way through the nervous system to get to a final destination. So they've got to go to the right place, or the whole brain doesn't work. So you've got to make all these connections. And often it's a series of intermediate steps that the growth cone has to take. It has to go to position A and then turn and grow to B and then turn and then grow to C and so forth in order to set up the pathway. As you must know, when you look at a brain, it is full of nerve pathways all over the place. It's really tangled and complicated. Who knows what's going where? Well, obviously the neurons have to know where they're going. So how do they know this sort of thing? And one of the models that people came up with was, well, clearly you've got a substrate. So you got things it's got to grow on. So, if you're doing this in the lab and you're doing it in a petri dish, the surface of the petri dish is your substrate. And the neurons have to be growing on this substrate and sensing what is there and using the information in the substrate to get to where they're going. We can do simple experiments with this where you can, for instance, put nerve growth factor, just take a drop of NGF, put it on one side of the petri dish, and it will diffuse. So it'll be high in concentration at one side and low in the other. And you throw some cultured neurons in there, and they will follow the gradient and get to their destination, which in this case is just the other side of the petri dish. So we can do that sort of thing. Another complication, though, is that when we look at the genetics of this system, they're complicated. It's not simply one factor and one cell following one factor. We get this mesh, this network of all these different fibers there. And furthermore, each of these neurons, as they're growing out, is laying down its own unique track. So before you know it, the nervous system gets really bewilderingly complex. There's all kinds of things going all over the place. And it's fairly easy to generate lots of complexity. There's lots of systems that will just spew out these connections, and you'll end up with a tangled network that works, but it's not clear how it gets there. One of the analogs here is think about neural nets and how neural nets can be be generated to generate specific outputs from a specific set of inputs. And most of the people working with neural nets have no idea what's going on in between. Same thing here in the brain. So what I was talking about in my video is one fairly simple system, which is the insect nervous system. And the insect nervous system starts off with a set of neuroblasts. Neuroblasts are just the progenitor cells that are going to produce the neurons. And so when you look in the early embryo, and in a grasshopper, you'd say when it's about 15% developed or thereabouts, what you've got is a floor that's made up of all these little bubbles of neuroblasts that are growing out. And each of those has its own unique signature, its own set of molecules that it expresses. So it starts off with this complex tapestry of molecular signals that the thing has to grow on. So that's one interesting thing. The paper I talked about was by Thomas Elkins and some others. And the cool thing about it is they had the goal of let's identify some of these markers, some of these little molecules that are sitting there that neurons can follow to get to their destination, and let's destroy them. So yeah, they use monoclonal antibody technology to identify a bunch of interesting molecules that are expressed in patterns on the surface of the nervous system. And then they used cool techniques that you have available in fruit flies to knock out those same genes, to delete them. And what they discovered is the first gene they were looking at was one they were very excited about. It's called fasciculin 1. It seems to follow into these very specific pathways. It's got a unique subset of pathways. We're all saying, oh, that's got to be really, really important. There's got to be something there to make sure that those neurons grow to the right place. And they killed the molecule and nothing. Not nothing, nothing, but something, nothing. That is, the nervous system grew out perfectly normally. So it's like you go out and kill this candidate molecule and the animal doesn't care. It can grow just fine. Well, that's mystifying. So there's all kinds of hypotheses you could make. Maybe that molecule is just coincidentally there. It's not really important in the stuff we're talking about. Another possibility was that maybe it's a cooperative thing. It's not just this fasciculin that's doing some work. It's some other gene that collaborates with. And so if you just knock out one, it doesn't make a big deal. You have to knock out two or more in order to get an effect. So anyway, what Thomas Elkins did was he found another molecule that's expressed in similar patterns, actually more general patterns, and knocked it out. That was the Abelson tyrosine kinase, which is a well-known gene involved in cancer and things like that. So you knock that one out, the animal develops fine. You knock out fasciculin 1, the animal develops fine. But you knock out fasciculin 1 and Abelson tyrosine kinase, and now you got a problem. Now it can't develop. In particular, it can't form the commissures that cross across the midline of the nervous system. I thought that was kind of an important discovery because it's saying, yeah, it's not just it's not like this simplistic model that many have people have that there is this hard-coded pathway that's all laid out and it's just like a paint by numbers kit. You got the cells just following along on these particular pathways to get where they're going. And no, that's not how it is at all. It's a little bit more artistic. These things have to negotiate a pathway that's complex and has redundant components and that interacts with other components in the nervous system. Okay, so I put that up. Uh, it's one of my least popular videos of all time. That's okay. I knew it was going to be a little bit obscure and specific to my interests. But it's an important point, I think, is that we have to get away from these reductionist ideas of how the nervous system develops. It's complicated. There's all kinds of stuff going on. And it's not like it's all hard coded. It's like you got to have these cells that grow out and they have to talk to each other. Okay, so that was my story. And part of the background for the story is that for many years I've been concerned about the reductionist approach many people take to genetics. That there's this idea that there is a gene for something. And you could get that in this Thomas Elkins paper too. There must be a gene for making a commissure. And no, there isn't. There's a couple of genes that have to work together. So genetics is more complicated than that. It's not reductionist. But the other part of the story I was trying to get out there is once you've got genetics, you got this other layer that comes along. You got the developmental layer, where you've got these cells that contain a particular genetic complement, and they have to interact with each other, and their interactions increase the complexity of the nervous system as they're sitting there negotiating with each other and responding to each other. So you got genetics as one layer that's not reductionist, and then you got development, which adds another layer of sophisticated to the whole system, sophistication of the whole system. And that's where I got one creationist comment that I thought I would just highlight. Not just to not just to be mean, but because I think it was important to recognize that some people still have this reductionist view of everything. Okay, so these are the comments that I got from this one guy whose name I'm not even going to try to pronounce. So he starts off with a little flattery as you were explaining this fascinating subject. I truly wonder how random mutation plus unguided selection could have produced such a complex information-rich system. He's made a bunch of mistakes here. What about selection is unguided? Selection is shaped by the environment, by the interactions it goes through. So drop that right away. You're already working with a misconception. And then he says, just imagine how much information is needed to code the right wiring pathways. I want to make it clear what's happening in development is that we are generating new information for the wiring pathways. It's not just all there all at once. It's the interactions between all these cells over time that produces these pathways. And then he says, I predict that there is no paper that show the formation of new functional neural pathways through random mutations. Yeah, actually, we do have those. There are lots of mutations that mess up the pathfinding, and so you end up with aberrant nervous systems. We've got lots of those in fruit flies. Uh, Things that, for instance, change the fate of some of the neuroblasts can produce all kinds of surprising effects. Okay, so he predicts there's no paper that show that, and I'll just tell you, yeah, I could find lots of papers to do that. Just look up anything about genetic manipulation of pathfinding. Yeah, we got them. They're there. And then he says this can only be explained by the hand of an intelligent designer. No, this is terrible. This is such a common tactic they take that they will undermine the argument by saying if it's not X, if it's not materialist scientific differentiation, it's got to be Y, supernatural intervention. No, there's all kinds of intermediates here, so that's bogus right there. And I gave him a few comments there, and he says later, I do believe in neural plasticity. Well, good. You got to accept neural plasticity because that's how we assemble this structure. As I've been saying, it's not hard coded from the get-go to be X and Y and Z. It's got to be worked out by negotiation over time between the cells. Okay, so he believes in neuroplasticity, but then he says it's a very controlled and guided phenomena that leads to memory and learning. I don't know where he got that. Okay. Uh, in the papers I was discussing, we're not at the stage where we worry about memory and learning yet. We're at the stage where we're trying to make fundamental connections between, for instance, sensory neurons and interneurons and motor neurons, just so we've got the wiring to work with. And then this is just descending into gobbledygook. What I meant was neural wiring by axons growth and pathfinding. Yeah, that's what I was talking about. To accomplish such a marvelous event, you absolutely need very precise coding morphogens. Okay, I had to stop there because morphogens has a very specific meaning, and I don't think he understands what it means. And it doesn't need very precise coding and so forth. What a morphogen is a substance that, by virtue of a gradient or other process that produces a differentiated levels of concentration across a region, that's all a morphogen is. And the kinds of things we were talking about in the grasshopper and the fly did not involve morphogens. They involved local interactions. Okay, so we already see that he's making stuff up. And fine-tuned ligand receptor concentration. Fine-tuned is a weird thing to throw in here. What does he mean by fine-tuned? Who's doing the fine-tuning? Generally, what we talk about when we talk about morphogens is gradients of concentration that are set up by distribution of enzymes or the distribution of things like the, the proteins and RNA that make that particular gradient. So classic example is bicoid in flies. It is generated because the fly embryo localizes bicoid RNA to one end of the animal. And then as that RNA is transcribed, it diffuses and generates the morphogen concentrations. So fine-tuned, I don't know. Anyway, then I told him that the event is not as rigid as he claimed and possessed a lot of high variability and neutrality of content, neutrality, but couldn't find a paper about it. I have my doubts that this guy is capable of finding a paper. But okay, we'll let it slide for now. Um, and then he comes back with, this third comment is just, I don't know whether he went to ChatGPT or something or found a paper and just pulled out a random chunk of it. So in contrast, every ganglion retina cell has a specific concentration of a chemical tag named F. And correspondingly on the tectum, there are ephrons, which are in contact with F signals and inhibitory message growth cone, which guide a general neural wiring. Okay, one of the things I have personally witnessed and seen is that the outgrowth of retinal growth cones to the tectum in things like amphibians, like frogs, you get this stream of growth cones that are moving across this flat sheet of the tectum, and they are making connections as they go. And yeah, then there's activity-dependent neural wiring, that there's various signals like you get visual signals into the eye that causes coded stimulation of the growth cones, and then they find a connection that matches. So there's all kinds of matching going on. But this has absolutely no relevance to what he was claiming. A precise neural network will form. He's really focused on this precision thing. Okay, that's fine. All right, let me see. What did I do with my stuff here? I just lost everything. Oh, there it is. I think. No. Oh, there it is. So let me stop sharing that. Okay, so yeah, he's trying to shoehorn it into this very precise, very systematic system. And yeah, you need some precision. You got to get things growing to the right place in general. And then there are mechanisms, mostly activity-based mechanisms, that help fine-tune and regulate how things are going to connect up. Uh, I just want to show you one other figure here that I really like. This is from Chris Do, and it's kind of a famous figure. Okay, what do you think of that? Very colorful, isn't it? What this is that Christo was studying neuroblast formation in Drosophila. And like I said, initially what you get is you get this sheet of cells that are on the ventral side of the nervous system. So you just get this sheet of these things growing out there. And what they are is a defined population of progenitor cells that will then subsequently divide to provide a more complex matrix for the nervous system. And so what you're seeing here is, if we go to the top left, that's at one particular stage. So we're looking at one early stage in Drosophila. And what you've got is just one, two, three, four, five, six, seven, eight, nine neuroblasts at that time. It starts out simple. This is one of the nice things about development is, yeah, you always go back and back and back until you find a simple system simple enough that you can understand it. So here we got just nine neuroblasts all sitting there. And what Chris Do did that was just amazing is he started using all these antibodies and other probes, RNA probes, for instance, to stain those cells for what kinds of molecules they're expressing. And look at there, they're not all the same. So over on the far right is the legend here, where you can see, for instance, oh, this bright red on S2 is for Fushitaratsu FUTS. It's a really famous segmental gene. And then we got wingless genes being switched on, and we've got huckabine and all these other very well-known transcription factors and receptors and ligands that are being expressed. And you can see a pattern here, and it's very consistent. So we got this pattern, and then what's going to happen is these neuroblasts, these big circles, are each going to divide a couple of times. And so the system will start getting complicated. So here a little bit later, we can see now we're up to 10 cells. We got this new one, 3,2, appearing up there, which has its own unique combination of molecules. And then we go to S2 and we've got some more divisions. And you see what's happening is as this process is going on, each of these cells is, as it's born, is looking around and expressing its own unique combination of various molecules in response to the environment. And so you see it gets messy pretty fast. So here at 5, you can see we got a whole bunch of cells here, and they're all expressing unique combinations of molecules on their surfaces. Well, in some cases, these are going to be cytoplasmic. But anyway, you get the idea, is everyone's unique, and they build this up pretty quickly. So this is an example of how this budding information can arise. And like I said earlier, this is a key thing. You're not starting with a precisely laid out series of informational molecules that predefine what you're going to build. No, as development proceeds, you're going to accumulate these and you're going to get all these different molecules expressed on the surface. It means the environment gets richer, the complexity increases, the information in the nervous system is steadily exploding as we add these new features. And not shown here is that these cells are also producing neurons. So they, at some point, they divide and produce a chain of neurons on their surface. And those neurons are then negotiating their pathways. They're growing out into this information-rich environment and finding new places to connect and building this wonderful nervous system that the fly or the grasshopper is going to use to locomote or sense its environment or mate or whatever it wants to do. So it's beautiful stuff. Okay, so this is Chris Doe's work. He's got a whole bunch of other stuff that he's done on these, but it's just iconic how we can now see with these molecular tools the diversity of substrates that are present in even a simple little baby grasshopper fruit fly nervous system. Now take that for a human. We've also got the same thing, a huge, rich, complex set of muscle neurons that are dividing and maturing in this system. So yeah, don't give me your reductionist nonsense. It just doesn't make sense. Okay. Uh, so let me just say one other thing about this. Um, that there are examples in vertebrates. I'm mostly used to working with invertebrates for this particular kind of thing, but then I had a long digression into zebrafish development. And zebrafish development is beautiful. It's got some really cool, complex stuff going on. But one other example I want to bring up is Ramachandran's work on synesthesia. Uh, Everyone's familiar with that, right? You've all read the papers because they're famous. They're really cool papers. Anyway, you know what synesthesia is? It's this phenomenon where some people make curious associations in their brain. That, for instance, for one person, the number five, whenever they see it in any context, is associated with red. And the number four is associated with yellow, etc. It's actually kind of useful. And it's powerful stuff if you've got this advantage where that means that you can pretty instantly detect concentrations or you can detect the identities of features in the environment because color is strongly associated with them. So you want to find all the fives in a diagram that's laid out in a piece of paper. For somebody like me who does not have synesthesia, I would look at that piece of paper and I just see a jumble of numbers all over the place. For somebody who's got this synesthesia trait, they would look at it and all the fives instantly light up as red. So they can quickly count all the red numbers, the red digits on the piece of paper. It's kind of a neat attribute, okay? And it's been tested. This is the kind of thing where you could say, well, maybe they're just lying and saying that red means five to them. Uh, but then you do those kinds of paper tests, those computer-based tests and so forth. And, it's just like with color blindness. You quickly detect uh, colors by using the test, and you can resolve these things. And it's kind of obvious that it works. So anyway, Ramachandran was doing all this work on synesthesia and particular between graphene and color. Graphene being shapes and numbers and letters and things like that, and finding people who had this association with color for these particular traits. Again, really cool stuff. I read it and I just wish I were a synesthete. A lot of you may be synesthetes already. It seems to be something that crops up a lot in artists. Anyway, so he's looking at all these sorts of things and he's trying to figure out what's going on. And what he did is he identified a region of the cerebral cortex where graphenes are encoded. And this is also really cool. There's a little patch in your brain that encodes numbers that floating around on the surface of the cortex There's a number one, a number two, a number three, a number four, etc. And fibers can just connect to that. And that's how you get the association of the digit five with the idea of five. So he found that little region in back here in the back of the brain. And then he noticed, well, many people noticed, it's right next to a color processing center. So there's a region where the projection from the retina gets sorted out into colors, color perception, and that's used to build the higher level colors that you understand. So we're talking about really low level stuff here, first of all. Anyway, how do synesthetes work? Well, it looks like they got really bushy terminal axons. So the axons are growing in to the region they're supposed to go. They're supposed to go to the color region and sort themselves out into the various colors, but they're not as precise as some people would have you think. They tend to branch a lot, and they have spuriously made connections into the graphene region. So you got this neuron that's supposed to be, it's supposed to sort out and form the pathways for sensing the color red, and it's also got this additional branch that happens to contact the digit five in their brain. And so they get this instant association between color and digit. So that's pretty cool. Um, this we think happens in everybody. That it's part of this activity dependent processing that allows us to prune and establish appropriate connections. So in boring people like me, who just have perfect eyes, uh, our retinas grew back to this region and they made their connection perfectly. They went to the color region. They said, this is where I'm supposed to be. I'm going to make a connection here. And no, I'm going to ignore all that spurious, nonsensical stuff about fives and fours and 11s or whatever. So, yeah, so my brain did everything just right, like it's supposed to. But in a synesthete, what seems to have happened is that they've made these cross connections. And normally, I shouldn't say normally, it's perfectly normal if you're a synesthete. Typically, uh, those connections get pruned as our visual system matures, not in these people. So they've still got this little light connection between color and numbers or digits, which is kind of neat. Doesn't necessarily give them any advantage. It's just one of those little spurious things that happens. So that was neat. But then he also did some other studies, and this kind of blew me away. He discovered that, there are drugs. Have you heard of this? There are drugs that meddle with your brain, that change things around in your brain. And one of the things he discovered is that certain hallucinogens unlock synesthesia. So it's saying that even in a mundane, boring person like me, where my connections are all as they should be, I probably have some fibers that have branched out into other areas where they should not be, ideally. But the process of fine-tuning the nervous system has disabled them. So we just said, okay, you've made a mistake there. You've taken a right turn and gone to the wrong part of the brain. We're not going to punish you. We're just going to silence you. We're going to just silence that region. So they've shut that down. But then if I go take the right hallucinogen, what may happen is it suddenly enables all these crazy connections. And part of the hallucination is I see colors in all kinds of unlikely, unexpected places. So I think that's the idea here is we have all these wild random connections all over the place in here. And what sorts them out is functionality. Do they actually contribute to the function we want for this part of the brain. And in synesthesia, it's one of those cases where it's got some function and maybe not important functions, and it tends to get pruned out. So anyway, that's the point is nervous systems are sloppier than you think. And part of the ways our brains work is not by intent, not by hard and fast rules. It's by these little random connections that we have the opportunity to find advantages for. So, if, for instance, there was intense selection for being able to sort out numbers, digits on a table, synesthesia becomes really useful. I just don't think that there's a huge selection pressure for that sort of function. Okay, so let me take a look at the comments here. We've got no questions here so far. Feel free, ask me questions about anything. It doesn't have to be about this brain stuff. It's just where my brain works lately. Uh, questions
Do we
about,
have any sense about whether synesthesia is genetically determined or can it be learned? Like if I
oh,
just looked at yellow fives all the time, would those neurons develop so that all fives would look like yellow?
mm, there is definitely a genetic component to it. Um, it doesn't seem to be a specific component. It's not like there's a gene that makes the nerves go connect between yellow and five. That just doesn't happen. What it more likely is if you've got if you've got a slacker brain that just says, "Yeah, get to this right area and find an appropriate connection and just hook up somewhere in there," that's going to produce synesthesia because it's going to tolerate more variation in the connections. But then other brains are going to be more specific, and you probably can't train to associate a particular color with a particular digit. Ramachandran has done this stuff as well, and what he finds is that there's a pattern, that there is a most common color associated with the most a particular graphene. And that's because we're looking at a spatial orientation, that these regions are next to each other. So it's just, if I use red five, I don't remember what the exact connections were. But if I say, okay, it's more likely for five to be associated with red, that's because the graphene map for five happens to place it near a region that's responsible for processing that particular color. So no, you probably can't train to get a particular thing. Could you possibly train your brain to be more synesthetic? I'm not going to say no. I'm going to say, yeah, it probably could be done. I don't know if anybody's done it. Yeah, your brains are pretty fluid. They can respond to all kinds of environmental signals. So I could see where maybe it could affect it somehow. Um, Go ahead, do the experiment. I would say start by looking at Ramachandran's papers and see what's associated with what, and then try to strengthen that association and see if it actually works. I don't know if it would. The impression I get from reading these papers is a lot of it is just that you make these spurious by chance connections, and that's what you're stuck with. Okay, anybody else have questions? I see Hemi is talking about owls. Oh, so the idea is, yes, some owls is speculated they can visualize sound when hunting prey under the snow, and that would not surprise me at all. Uh, that you could have these connections um, selected for, so they don't, so that a particular, a mouse squeak is associated with something particularly vibrant and easy to remember. Okay, I see my creationist has showed up in the comments. He says, so by that examples, you are going to deny the modularity of the brain? What the? No, brains are modular. There is a lot of modularity there. Why would I deny the modularity of the brain? Yeah, give me an intelligent question, somebody. I hate wasting my time with creationists who don't know anything they're talking about. As I said, you could ask me anything. You were supposed to ask me about my knee. Oh, yeah, I forgot. So I've been really slow getting back to things this past week because my knee went kerboom. Um, and so I've been kind of laid up for the last week or so, struggling. It's really boring to lie in bed for very long, but it's paying off because things are healing. The pain is going away. Uh, this is another thing biology is really good at is adapting to circumstances. And so, yeah, there's been some improvement on the knee front, which is always good to see. Good to see. I go in for an MRI on Tuesday, and because the pain has gone down so much, I'm kind of hoping that what they're going to tell me is, no, we don't need to take a scalpel to your knee. We're just going to have you go through all kinds of physical therapy. Oh. We'll get back to IgLU phylogeny in just a moment. Let me just finish. Ollie Polly, is there anything about the knee that you learned since you talked about it last? No, it's a total mystery. Mystery. Knees are just weird things. I've spent some time looking at the anatomy of all these tendons and the cartilage bits there, and I don't know how we got something so messy. Must have been an intelligent designer that put it all there, right? No, I don't. You look at it, and it's just kind of clear that it's kind of put together, like higgledy-piggledy, and we got something functional, and that's what we've got. Oh, okay, we got a couple more questions. Are there any theories regarding why there is redundancy in the genes that guide neural development? Uh, that's just standard evolutionary theory. Yeah, we don't want a single gene mutation to cause a catastrophic failure, especially in organisms like fruit flies that are such rapid developing, prolific animals that you don't really want to tolerate errors that mess up the whole system. So having redundancy is a beautiful way to simplify it so that you can at least get some offspring out of this that can then have sex with another fly and by recombination repair that particular structure. You don't want all your progeny to be total failures. So yeah, redundancy makes sense that way. Okay. Uh, then this is a curious question. Are vertebrate brains so large because of their inverted retina? Hmm. I don't see how that would relate. So, the eye evolves by an outpoaching of a portion of the brain. So the brain actually just kind of grows out and makes a little bulge and then gets an indentation and then we get the other lens in there and everything. And so that portion is going to form the retina. And I don't think that would be related to the size of the brain because there are lots of vertebrate, very tiny vertebrates who have the same arrangement in their retina. Okay, so our creationist asks another question, which is vaguely interesting. If all vertebrates are related to each other, why then is there so much diversity in the basement of development, like cleavage, gastrulation, urulation, etc.? Um, what? So much diversity in the basement of development. Okay, if we just think about something like gastrulation, um, gastrulation is a common process that fish, frogs, mice, people, chickens, they've all got to go through gastrulation, which is this process where we take a two-layered animal and we fold portions of it in to make a third layer. So that's how you get from ectoderm and endoderm to mesoderm in addition. Um, and what do you mean by so much diversity in the because if, for instance, if you look at gastrulation, we're all using the same genes. You know, that we're all setting up the same patterns using the same genes. The differences are topological, not even topological. They're still topologically similar. It's just that, for instance, in humans, we have a blasted disc. We have a sheet that's going to do this whole business. And if I look at a zebrafish, it's not, it starts off as a sheet on top of the yolk, but the yolk is so huge that it displaces everything. And so then we get this little cap of cells that have to do this migration into the interior. Um, but gastrulation in a zebrafish and a human and a frog look very different, but when you actually look at the molecules and the movements that are going on, they're actually closely related to each other. So, hmm, okay. Isn't neural crest development a mammalian brain development commonality? No, it's not. It's not exclusively mammalian. A lot of the work on neural crest has been done in salamanders, for instance. And birds, of course, also go through neural crest development. Uh, the common name used for these now is set aside cells. That what we see in development, it's another example of redundancy and the plurality of mechanisms, is that we see lots of examples where organisms early in development are putting things together, and maybe there's something that doesn't quite fit in, that doesn't function as well. We just sort of set those aside in evolutionary history. So they're just sitting there. You can imagine that in many cases, you get, okay, we made a bunch of extra cells. They don't hurt anything. We'll just leave them lying around. And so lots of organisms have these set aside cells. In the case of mammals and birds and things, yeah, actually, most of the work I know of in neural crest has been done in birds. Birds are really good for this particular topic. Anyway, so what's happening is you got these set aside cells that are just kind of sitting there in many of the older orders, maybe, but in some organisms, they have taken advantage of these. And then they've switched on functionality, added new functionality. We've got mutations that generate interesting patterns. And for example, in vertebrates, the vertebrates that have complex nervous systems, like us, one of the features of neural crest is to take these cells and wrap them around the peripheral nerves to make a myelin sheath to improve the speed of conduction. So you get faster responses in the environment. And that was done by just taking some of these leftover cells, acquiring mutations that associate them with the nervous system, and wrapping those around and making that insulation for the peripheral nerves. So yeah, it's also involved in mammalian brain development, also bird brain, yeah, everything. Lots of vertebrates use this stuff. Um, so there's different populations of these cells. There's the classic neural crest, which will arise, as you might guess from the name, along the crest of the nervous system, the dorsalmost portion of the spinal cord, that then migrate out and populate all the other peripheral regions of the body. But there's also a big pool of neural crest-like cells. I don't think they're called neural crests. They got a different name. But anyway, another population of cells in the brain that then migrate over the surface of the brain, and they contribute to things like um, auditory system, also visual system. All these things we really require the neural crest to do that. One of the things I've often told my students is that if you didn't have any neural crest, basically your face would be gone from about here down. Oh, a creationist is back. How can we combine evolutionary theory with developmental biology when we only see loss of function types of mutations in developmental genes? Where do you get that from? We got all kinds of examples of gain of function we got novel mutations. I just mentioned one. The neural crest genes, neural crest cells have acquired new functions in evolution to enable more complex morphologies, to encode more complex behaviors, for instance, add new functionality to the nervous system. Uh, Loss of function types of mutations in development. No, there's lots of other things. This is what happens when you come in with no background and you engage in some motivated reasoning to say, this is the way it must be in order to make my worldview work, instead of looking at the evidence. Okay, we're getting about to the time. Larry's going back to Azeroth. Oh, I haven't played that in so long. One of the things that frustrated me about that game was all once you get into it and you start playing, the NPCs have such fixed functions, and it got to the point where it was driving me crazy to have some you know, goofy bird creature saying the same thing to me every day when I logged in. So, yeah, I have a low tolerance for that kind of thing, I guess. Okay, unless there are other questions, I think we can wrap it up here. That was fun. Should I do this again in the future? Maybe. As I mentioned at the beginning, I do have this Patreon account. Patreon.com/PZMyers. Eric is a member of that. Uh, it's I've really taken a low bar to that, and I say, okay, you can just for $1 a month or up to $5 a month, you can join, and then I post some things there. I got to post more spider pictures there, but it's little things like all my videos now. I post ad-free to Patreon first the day before I release them on YouTube, where they get muddled up with all kinds of ads. One last question. How smart are dolphins? I don't know. What is smart? This is a big question. What does intelligence mean? Are dolphins intelligent? Yes, they are, but so are spiders. Everything is intelligent for its particular functions. So I don't know. We often bias it by thinking along the lines of how close to human intelligence are they? Yeah, I don't. Okay, dolphins are pretty smart, but possibly not as smart as cephalopods. I don't know. Um, the estimates I've seen of neural function in cephalopods suggest that they have about the neuronal equivalent of a dog, in terms of number of neurons. But then it's really hard to make those comparisons because Um, One of the things about vertebrate brains, vertebrate nervous systems, is that we've got this very cheap mentality to our nervous system where we just make lots of little fine, very general sorts of neurons all over the place. And then they get funneled into their functional regions, and that's where the smartness comes out of them. Uh, Things like cephalopods and insects have far smaller numbers of neurons, but those neurons are beautifully fine-tuned and have all kinds of functions to them that are encoded in their structure. They're not just cables, right? They have all kinds of different pathways, and they have all kinds of different receptors and signals scattered along their surface, and they do very complex sorts of things. Oh God, Rashad is back. What's your thought about orphan genes? Aren't they a challenge for evolution? No. Um, Orphan genes are really routine. There's a small number of them, and all it takes to make a patch of DNA express itself as a protein is you just have to tag the right kind of markers around it. And then it gets expressed, and then it gets processed, and then interesting things happen. So, okay, Hemi, you're getting a weird mystery funhouse distortion to the side. Yeah, that's because, yeah, I'm using Zoom's function to blur the background because, and it gets kind of funky at times. Yeah. Oh, look at the top of the hairs at the top of my head. I can make them disappear and reappear. Oh, yeah. It's just an artifact of Zoom. Okay, I'm gonna stop there. You don't want to hear me babbling all day long. It's almost 4 o'clock, so it's been about an hour. Perfect. I will try to do this again in the future, and feel free to send me questions. Feel free to send me ideas of things I should talk about. I do have a little bit of a background in the nervous system and both function and development, and I'm primarily interested in developmental biology. Ooh. Okay, Adrian, stop asking interesting questions. Are there evolutionary arms races of intelligence? Yeah, maybe we can save that for the next time. Okay. Talk to y'all later. Thanks for stopping by. It's been fun. And I guess I will turn this off. Bye.